right, ladies and gentlemen, I don't know how many of you were here last week, but I suspect most of you. We're coming in on the middle of a lesson. So if you weren't here, I'm sorry about that. I don't have time to teach first lesson 1 again. So we're going to pick up where we left off. And what I've been doing the last 2 or 3 days, since I really didn't get as far as I wanted to last week, is redoing 2 lessons into 1. So we'll see how that works. So if I get to speaking at 150 miles an hour, you'll understand. But it's a great thing to have you here tonight, and it's a joy to share with you things that I have learned about God's world that have been absolutely, overwhelmingly faith-building. My goal is to help you be a stronger believer in the God who made you and to absolutely learn how magnificent He is. And if you don't understand everything we talk about tonight, you are forgiven. Try to get as much as you can, but mainly get the big picture of what it's all about. So, tonight, our topic is going to relate to cells and molecular machines. That was my lesson design for tonight, and what I've done is reorganize it for you, and including a little bit of the end of last night's. So welcome. Let me just say a couple of words of thanksgiving to the elders for providing this occasion, and I understand all summer you're doing special lessons on Wednesday nights, and God bless you for that. I hope the collective of those series together will help you in every way. And I appreciate the fact that Brent and Hayden are working together here in the Lord's kingdom, and wish them God's blessings. And we know Brent and Tamara from way back, before they got married, and even before that, sometime. So we love them, and we appreciate what they do for the Lord's sake. And glad to be with you. Thank you for the opportunity. And that's all the time I'm going to take on that, because we need to get busy here. So I want to start tonight with the same place we started last time, because when I do this series in a church building, it is fitting in my mind to start with a passage of Scripture. And I'm trying to show folks that what I'm doing tonight is supported by scriptural teaching. So Romans 1:20 is the key passage we're going to look at again this evening, which says the following: "For since the creation of the world, God's invisible attributes are clearly seen, being understood by the things that are made, even His eternal power and Godhead, so that they are without excuse." What that passage teaches is you can see an invisible God by seeing the things that are made, and they testify of a glorious, grand, majestic, powerful, and even not of this nature. He's divine. He's not a part of nature. That you can see from examining the things that are made. It took something beyond nature to produce what we're going to talk about tonight. And so it's the famous argument from design. I believe that's always been the strongest argument for the existence of God from the natural world. Design is everywhere. So I want to offer an apology again at the beginning tonight. When I get finished, I will not have touched the veritable hem of the garment of what needs to be said. So please forgive me from the beginning. We'll just barely touch the hem of the garment. But I hope in the process you'll be overwhelmed with the majesty of God. All right, so that's the biblical basis for what we're doing. And for the rest of my time tonight, until the invitation way at the end, I'm not going to use this book. This is God's book, and I love preaching from it, and it's the most important book that's ever been made, and it is unique in all time, and the evidence to support that it's God's Word is overwhelming. But that's another series of lessons. We'll let Brent and Hayden do those. I'm going to turn instead to God's other book, the book of the natural world. So welcome back to biochemistry class number 2. You missed class 1. I'm going to give you a test anyway. So let me just ask you a few things, folks, and see what you remember. We did a little basics last night about biochemistry, didn't we? The 2 great kinds of molecules in every living thing, One is, don't tell me you're going to miss this one. Proteins. Yes. Proteins are vitally important, but there's another great class called amino acids make up the proteins. It's nucleic acids. It's called DNA and RNA, if you remember. Every living thing has those two things in it. And what we spent most of our time on last time was how do you get proteins? Remember? So we'll just review that a little bit. We didn't spend as much time on DNA, and we really need to give it more credence, but it's all there. So let's start this way tonight. Had you lived in the early 1800s and you attended university, you would be required to take a course in natural theology. I want you to notice the subheading to this famous book by William Payne, or Evidences of the Existence and Attributes of the Deity Collected from the Appearances of Nature. The whole course was to show you that you look at nature, everywhere you look, you see evidences for God. That's what the course was about. You probably don't know that Charles Darwin took this course on natural theology, and he studied the book by William Payne, which was the most famous book of that century. So William Payne is an outstanding example of someone who used evidence from the natural world to convince people there was a God. And his famous argument, called the watchmaker argument, you've probably heard. He started his class that way. And I'm tempted to speak in British and do that with you, but I don't want to take the time for that. But he would say, if you went along a heath and you stumbled over a rock and you kicked it over and there you found a clock or a watch, probably the first thing you would think is not, isn't it interesting how that just evolved or came to be? You would probably think, where's the guy who made that? I need to find him, right? Or somebody bought this from somebody who made it. I mean, that's the logical thing you'd think if you found a watch. So that was the beginning of his book. Well, what I want to do tonight is I want to show you what Richard Dawkins does with that. Richard Dawkins is the professor of the public knowledge of science at Oxford University. He's been given that title because they think he's one of the best proponents of how to let people know how to treat science and how to learn what science has to tell us. I think he's one of the worst. And so he wrote a book in response to Dr. Paley's book called The Blind Watchmaker. So I want to share you a little bit from that book. By the way, I have a copy of Natural Theology if you're interested. There's a modern day version of Paley's book. It's worth your read, even 150 years later. And then here's Richard Dawkins. I bring these books with me so you can come up here and take pictures if you want. And I'm going to send you. Did I send you a bibliography? Okay, well, you'll get it soon enough. So I want to just read you a little bit about Dawkins' introduction. We animals are the most complicated things in the known universe. Does that give you an idea where he's coming from? Biology is the study of complicated things that give the appearance of having been designed for a purpose. He admits in the first page, everywhere you look, things look like they were designed for a purpose. And then, ladies and gentlemen, he spends 300 pages trying to convince you they were not designed. But he admits they sure look like they were designed. So when the Bible says, when you examine the things in nature that are made, God's serious about that. And everywhere you look, that's what it looks like. And why not take the obvious conclusion, yes, those were made. No, not these guys. So I'm going to turn over now, a little bit later in this first chapter. The watchmaker of my title is borrowed from a famous treatise by the 18th century theologian William Payne. His Natural Theology, published in 1802, is the best known exposition of the argument from design, always the most influential of the arguments for the existence of God. It is a book I greatly admire, for in his own time, its author succeeded in what I am trying to do now. He had a point to make. He passionately believed it. And he spared no effort to ram it home clearly. So I always like to read that for you, class, because I yell sometimes. And I make no apology for that. Because the other side of this argument is presented all the time in the public media, everywhere you look, trying to say that these things were not designed. And frankly, I get quite upset about that. And you need to hear the other side, and you need to hear it forcefully. So I also have a point to make. I passionately believe in it, and I make no effort not to ram it home. Then he goes on to say this. Paley's argument is made with passionate sincerity and is informed by the best biological scholarship of his day, but it is wrong. Gloriously and utterly wrong. The analogy between a telescope and an eye, between a watch and a living organism, is false. All appearances to the contrary, let's put it up here. All appearances to the contrary, the only watchmaker in nature is the blind forces of physics, albeit deployed in a very special way. Did you get that, class? All you need, folks, is the blind forces of physics acted on by natural selection. That's all you need. And it explains everything. So this gentleman said. And he proceeds to say, a true watchmaker has foresight. He designs his cogs and springs and plans their interconnections with a future purpose in his mind's eye. Natural selection, the blind, unconscious, automatic process which Darwin discovered and which we now know is the explanation for the existence and apparent purposeful form of all life, has no purpose. It has no mind, no mind's eye. It does not plan for the future. It has no vision, no foresight, no sight at all. If it can be said to play the role of a watchmaker in nature, it is the blind watchmaker. That's where he gets his title. Did I lose you? He's saying the blind forces of physics acted on by natural selection accounts for everything you see in the living world. And it's the biggest lie that's been told ever. It's a fairy tale. But he believes it with all his heart and tries to propound it to you folks. So let's talk a little bit about what Darwin actually said. I didn't bring his book with me tonight, but his book, On the Origin of Species, is probably the most life-changing text that's ever been written next to only the Bible. Because its view of the world and how it came about has changed the way we think about almost everything. But one thing he did in that book repeatedly was critique his own theory. So here's one. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous, watch this, look at the screen, numerous, successive, slight modifications, my theory would absolutely break down. Listen to me closely, class. Darwin said if you're going to get from here to something really complex, you've got to do it in small steps, which add up over long periods of time. You see what he says? Numerous, successive, slight modifications. Darwin would say to you, if he were teaching this class tonight, he would say, nature abhors leaps. Nature doesn't have a bird lay an egg and it comes out an elephant. That doesn't happen. But he would say that creatures over time can change a little bit and then a little bit and then a little bit, numerous successive modifications over long periods of time, and you can produce anything as long as you break it down into small enough steps. And folks, that is precisely what Dawkins does in this book. He is the ultimate reductionist. If something's complicated, then break it down into parts that are not complicated. And if that's still too complicated, break it down more. And if you break it down far enough, one of those simple steps could have happened without any God or creator. And then you add another simple step, you get a little more complicated, and so on. But that's a very important thing. So could we produce such a thing? Well, in the first place, it's hard to prove a negative. So the way he wrote this is a little tricky. If it demonstrated that there's any complex one which could not possibly have been formed, well, I will tell you, these guys are brilliant in making up stories about how it might have happened with very little evidence to support it. But that's what he said. He believes he answered it. I would present to him this organ right here, the cell. And I will tell you, in the 1850s, when Darwin first wrote his book, they didn't think the cell was much. A little blob of albuminous carbon, just a blob. It was so simple that surely that could have been formed by natural causes. You do know, don't you, that many people believe that living things came from rotten meat by itself? There was a very common conception that living things were produced from non-living things naturally all the time. Who was it that disproved that in the 1800s? Do you remember? Famous, known for milk. Pasteur. Pasteur's experiments demonstrated living things always come from living things. Period. But Dawkins would say, if it's living now, it didn't have to be before it got to be living. It could have come about by small steps over long periods of time. I say, let's talk about the cell. We've learned one thing so far, class, that's important to my lesson. We talked about how you build one protein. You remember this? I'm going to test your memory a little bit. The process of building one protein inside every cell in your body starts up here in the nucleus. Whoops, let's go back there. Starts up here in the nucleus with the DNA. The DNA has all the information in it, you remember? But it can't copy itself. It can't reproduce itself. It needs what, class? Proteins. Now get ready to give me an answer, okay? You first have to unzip the DNA. I'm just going to run through this briefly. This process is called transcription. You're copying the information off of the DNA onto an RNA. You're making a copy. It's transcription. Well, what does it take, class, to make DNA unzip? Protein. Good. And then you have to read it, and then you have to zip it back. What do you think it takes to zip it back? Protein. Good. Now, there's a whole lot more to that story right here, as I'll get to in just a minute. There's more than those number of proteins. There's a whole lot more going on. Once the copy is made, you have messenger RNA, ribosomal RNA, and transfer RNA all being made. We're not even going to talk about these over here, but believe me, there's a long story about both of those that is very complicated. And we're not even touching it. We're talking about this one. You see right here, it's got some white spots in there. Those are called introns. To mature this transcript right here, you have to snip out those introns and mature it. That's what that's called. Guess what it takes to make that happen, class? Proteins. A bunch of proteins are involved, as we'll get back to in a minute, but there's proteins involved there. Then it has to migrate outside the nucleus through those holes. You remember this? It has to go through the hole. What do you think guards the hole, class? Proteins. Can you see right there, there's a little thing to show you there's a channel? Every one of those holes has a channel. There are hundreds of proteins in that channel. And those are required to allow that migration to take place. You with me? Migration, class, requires a bunch of proteins. And the more we study it, the more it takes, or the more we learn about. Okay, and then in addition, once it's migrated outside the nucleus, it comes and picks up by two ducks hugging each other, right? Those are ribosomes. You have hundreds of them in every cell of your body. Hundreds of them. Their job is to take this information, read it, that's called translation, and produce proteins. And producing proteins means you're lining up a bunch of amino acids in a row. You remember this? Class, a string of amino acids is a protein. That was weak. Let's get a little more enthusiastic. From the beginning, you read at methionine to the end to a stop message. There are 3-letter words that stand for amino acids, and a chain of amino acids is a protein. Exactly, and the code for that protein came from up here through this long process and is finally produced down here after you get through the ribosomes. What I didn't tell you last time, because I got rudely interrupted at the end of my lesson and had to stop, is that after that translation takes place, there's a whole bunch of other stuff that has to happen next that's not on this picture at all. There's a whole bunch of other things, which I'll talk more briefly about in just a moment. That was our review, class. Every step of building one protein required what? Proteins. My goal in a few minutes is to tell you how many at this stage in our understanding it requires, how many proteins to build one. And I want to make this for the first time now tonight. Don't miss this. Every one of the proteins required to build one protein have to be built the same way. Did you get it? Let me say it again. It takes many proteins to build this one right here. This guy you're ending up with down here. It took all those proteins to make that happen. But every one of those proteins has to be built the same way. So how did you get them? Let's go back here to the beginning. To unzip DNA requires a specific enzyme, which is a protein. You can't even unzip it without that. So how'd you get that first protein to unzip it when it takes that protein to unzip it to make the process start? Am I getting this across? It's like the chicken and the egg problem, isn't it? The fact is, class, anybody with common sense can think through this. You have to have all of this or you get no proteins. And if you get no proteins, you have no life. Right? Now, here's what I want to do next, which also I was planning last night. Drew Berry is one of the best illustrators I know of. He lives in Australia, and his life's goal is to make things come alive to you that you can't see. They're so small. So he's done a lot of fascinating videos. You can go to his website and have a lot of fun with that if you want to do that sometime. So I'm going to show you one of his videos. And Hayden, I need to pause right here and tell you that Hayden is my champion, because he helped me get ready for this and came over here and practiced with me, because his job for the next few minutes as we show videos is to start and stop that video at my command instantaneously. And if he messes up, it only ruins my whole speech, so there's no pressure on him whatsoever. No, he's really good. But it does help me not to have to do that. So here is the video clip, and what this is, ladies and gentlemen, if you want to go ahead and start it just briefly. Stop right there. This is the start of a gene for a bone strength protein. You're getting ready to build a bone strength protein. You know you need proteins for your bone strength. Well, this is how you build one, according to his little visual. And it's kind of interesting because, look, this right here is the DNA. It's like that strand that's coiled. These are globs of, what do you think, class? Yes, proteins. And he colors them, not because they are that color, but to make you notice their differences. That's one protein. Can you see in the background there's a little bit of one back there. Here's another one. Here's another one. That's several different proteins, not just one. Okay, let's watch now for a little while. In a minute, we're going to activate that group of proteins. You see the little glob? Stop right there. Oops, we missed him. Go a little farther. There. Now it's behind the vitamin. See, that's a vitamin D, class. And he's going to join up with this protein right here. Go ahead. Which activates him. Stop. That turns him green. No, it does not. It just makes him go. So he likes to turn things from red to green because that means go. All right, so here you go. You got an activated protein glob. which now gets busy. Let's watch. Activated by vitamin D. So you get out in your sun in Florida and you're good. Here's a bunch of more what? Proteins. Stop right there. This specialized protein is called RNA polymerase. If a word in biochemistry ends in ASE, it is an enzyme. And an enzyme is a special case of a protein. Thank you very much. That's another protein. So the RNA polymerase actually rides along the DNA, as you will see here. Go ahead. And as it does so, it reads the information and then starts producing the messenger RNA. This is the messenger RNA. This is, stop right there. The DNA is moving this way. The RNA is being produced down in this glob of what? Proteins, and then it's being released out into the cytoplasm, and that's the RNA. That's the copy of the information. Go. So we'll watch it a little while here. See all this junk floating around on the outside? That is not junk, class. That's critically important material for building RNA. And it's going to go down inside this glob, as you'll see in a little bit. Watch there now. See all that stuff going in? Stop right there. We're now down inside, and can you see it's unzipped? We unzipped the DNA, and notice what's happening. You've got exposure of this side here and of that side, and we used to think only this side was copied. Now we know very well that many times both sides are copied, and a whole lot of other things happening we didn't know before. But it's making copies of this exposed side. Go ahead now. Okay, stop right there. Can you see these three-letter words? G, T, A, G, C, and then it goes right on. Each three-letter word stands for a specific amino acid. Remember? There's 61 of them, 61 specific codes for the 20 common amino acids in every protein and every living thing. It's coded right here. So it's making copies of that code, class. Right? Are you with me? It's doing it one at a time. If there's an A over here, there's going to be a T over there. If there's a G over here, there's going to be a C over there. If there's a C over here, there'll be a G over there, and so on. It's the exact mirror image. Now let's watch just a little bit. Can you see they're coming up there one at a time? Boom, 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 boom. Stop. One of the problems, class, with previous videos that I've used is they don't show you how rapidly this takes place. May I tell you, class, that the average protein in a living cell is 1,000 units long. 1,000 amino acids. That means you've got to add 1,000 of those in a row. That can be done in microseconds. And the videos didn't show that. So his goal in this video is to show you how rapidly this is taking place. So here we go. That's real-time speed, class, for copying to make a protein. Now stop right there just a second. So we've talked about transcription down in here, making this copy. That's only the first step, right? And I'm sorry, but in his video, he skips the whole migration thing. He doesn't even talk about that. Sorry. So I'm going to go on. My only thing now for the rest of this time on this one is to show you how fast this happens. So let's go. So he releases the messenger RNA into the cytoplasm. It has to migrate. You see it coming out of the hole right there into the nuclear protoplasm? And it's picked up by the ribosome. Here it comes. Boom. Stop right there just a second. A ribosome, class, is made up of RNA and what else do you think? Protein. 90 different proteins. Did you catch that? I should have had some of you adding up how many times I said numbers here. But I'm going to come back to the total numbers. RNA has at least 90 different proteins plus the RNA in it to do what it does to help build a protein. 90 distinct proteins that each have their own task. Let's keep going. How does it do it? It's got to read the code here. Can you see there's little three-letter words here? Well, it's gone. What's happening now is this is the blob of ribosome. These are transfer RNAs. They're bringing in the appropriate amino acids. Here's one blown up for you. These guys look a little bit like seahorses, I think. And they're coming up inside here, and they're bringing up the appropriate amino acids. In just a minute, we're going to watch this as it shows you several. So watch with me here. Keep your eyes glued up here. There's 1, 2, 3, 4, 5, 6. That's 6, 7 different ones. How many amino acids do you remember? Total of 20. That's the first thing I presented to you. So there are 20 different transfer RNAs, and each one of those has a specific, what do you think? Yes, the answer is always protein class. Each transfer RNA has to have a specific protein to go with it, or it doesn't work. That's 20 more proteins. Each specific for its task. If you give it a transfer RNA, you give it a protein that goes with a wrong amino acid, it doesn't work. I keep saying this, class. This has all been designed. This didn't just come about by natural causes. Let's keep going. So it's reading the 3-letter codes, and down inside is the action. So let's stop right there just a second. There's the transfer RNA. At this end, it has the code that matches that 3-letter code. At this end, it's got the appropriate amino acid, and it's being added one at a time to this strand right here. This is making hemoglobin this time. So that's why it's colored red. Let's go and watch it a little bit. Now what he's going to do here is try to speed it up a little bit and show you how rapidly this really happens. This is way too slow. Move it. See the thing forming up here? Stop right there just a second. Well, that's okay. Go ahead. Right there, yeah, good. How'd you do that? He's quick. What I want you to notice is the code on the transfer RNA is down here at this end. The amino acid is up there. The amino acid never touches this code. It's the transfer RNA that manages that. You ought to be saying, oh, it's amazing. Keep going. Every step in this process is critically important, and every single step requires more, what do you think? Proteins. There's a bunch of other proteins happening while all that's taking place, and now we're finally getting ready for a finalized string of amino acids, which is a protein. But class, a string of amino acids isn't good enough. Just having a string of amino acids will get you nothing. Every one of those proteins has to fold up into a specific 3-dimensional shape or it does not work. It's called the folding phenomenon. Guess what it takes, class, to make proteins properly fold? Protein. More proteins. That's part of the translation process that has to start going as soon as it's made. You got to fold it. That thing is not finished. Keep going and let's finish it now. There it sits. It's not finished. And he doesn't do anything with that in this video, I'm sorry to say. But there's a bunch of post-translation stuff that goes on that's got to be done before you have a functional protein. And it's a bunch more proteins, as you'll see in a minute. Okay, that's the end of that one, right? So we go on. Now let's go back to this a minute. Copying the messenger RNA, then it has to be matured. You know what evolutionists started saying about these introns, those little things you kind of snip out? They were saying, why would God make something where you're going to copy it and then you're going to use it down here to translate and build a protein? Why would he make something that you have to mature before you can use it? That's stupid. And by the way, what happens with these introns? You chop them out and throw them away. They're just junk. What they would say to you, class, is that's a testimonial that this came about by a slow evolutionary process. Nobody was guiding this. No smart God would do that. And so they began to call a lot of the DNA junk because it's not doing anything. It seems to be useless. And class, from about the late 90s on up to almost 2010, there were books written, Richard Dawkins in the middle of it, saying things like this. Well, I don't even have that. I skipped that, sorry. Let's go back. But Dawkins said this. Only about 5% of the DNA up there at the top is being used to build proteins. The other 95% might as well not even be there for all the good it does. So it began to be propounded that 95% of DNA was junk. And you creationists better get over it, because God wouldn't make junk, would he? This is a better evidence for evolution than it is for God, because you got 95% junk. Now, class, it is the case that less than 5% of the DNA is actually used to build proteins, which is utterly amazing because there are thousands of them. So what's the rest of it doing? Good question. But you know what a creationist says about that? It's doing something, we just don't know yet what it's doing. And you better be a little careful about calling things junk when you know not what you're talking about. And I'm happy to tell you that Dr. Jonathan Wells, wrote a book recently called The Myth of Junk DNA. And it's full of declarations of what that other junk is doing, which I don't have time to tell you about. But there's a lot of things going on inside that cell that we knew nothing about 10 years ago. And it's utterly incredible. Don't you dare call it junk. And I heard a public apology from Richard Dawkins for his stupid statements as the professor of public sciences? Not a word. But he knows it ain't so. So what have we learned from some of these studies? There's a whole program after they did the Human Genome Project. You remember that? That was from 1990 to 2003, and we mapped the whole human genome. They found lots of things about that disturbing. I didn't know how to answer, I will tell you that. A lot of it looked like it was junk. But you know my answer to that nowadays? Wait about 20 years till we can find out what more we can learn about the supposed junk. And one thing we've learned is it's much more densely concentrated than we ever dreamed, because multiple messages are stored in the same sequence of bases. You get that? Let's go back to that introns. You know what we've learned, class? When you snip these things out, you have to re-edit them and put them back together. Who says you have to put them back in that order? If you put them back in that order, you've got one transcript here to make one particular protein. What if you put this one first, that one second, that one third, and that one fourth in your mature? You got a different protein. You with me, class? If you have a different string of amino acids, you got a different protein. What if you put them this one and then skip to that one and come back to that one? What if you had more introns? Class, did you know we have now discovered that one section of the genome that's been copied can make up to 1,000 different proteins by this snipping and editing process? So that one piece of the genome had about 1,000 more times information in it than we knew before. And those introns, far from being junk, are used to make little RNAs and do all kinds of important stuff in your body, and if you don't have them, you're dead. Call it junk if you like. I repeat what I said last night. Don't you believe what I'm saying just because I'm saying it. You read these books for yourself. That's the first bell. Now, you promised me last night that we'll come back and finish this up. But I want to do something else. Let me see how far I can get. Spliceosomes. Let's at least get that far. You remember I told you they had proteins to splice it? There's a picture of a spliceosome. Folks, we know all about spliceosomes. No, I take that back. We have learned a lot about spliceosomes. There's still a lot to learn. Spliceosomes are several different SNRMPs joined with other proteins to form a spliceosome. Class, in this blob of spliceosomes, you see those blobs there that come together? Among those blobs, there are over 150 different proteins being used at the same time to manage that process. Over 150. Just in that one thing. That's the guy that goes in there and snips. See, this is the original intron up here. It snips it, closes it up, it snips it off, and then it closes it up and then snips off this end and puts the exons back together. That's a complex process. The chemistry is really complicated. It takes over 150 proteins to accomplish that, class. That's in that one step of maturing. That's another 150 to add to your number that are required. So I think I will close with this. You remember this? How many proteins does it take approximately for this transcription process up here, which involves copying, transcription, it involves maturing, and then you have to, of course, migrate it. We'll get to that in a minute. How many do you think it takes for that? At least 232 different proteins just for that. That's what we know about today. How about migration? At least 41 different proteins are required to help this little guy migrate outside that hole. 30 of those are at the hole right there. What about translation down here? I'm talking about when you translate and make a protein and all the stuff that happens afterwards. It's called post-translational activities. There are lots of them. I have to show you this. I'll put together a document that is 7 pages long. It's called The Number of Proteins Needed to Make One Protein. It's interesting reading. It is now a total of 7 pages long with all kinds of detail in it about how many proteins are needed. So what's the grand total? Well, to do the post-translation requires 1,854 different proteins. Now add those up real quick in your head, would you? I've done it for you. There's a grand total of at least 2,127 different proteins required to make one protein. And every one of those has to be built the same way. Now, ladies and gentlemen of the jury, I hope I wouldn't need much more to convince you. Somebody designed that. That did not come about by natural causes, small changes over millions of years. Are you kidding? Something would have passed out and passed and not even been formed long before anything useful came about. So one of the great discoveries of the scientific 20th and 21st centuries is that even the simplest forms of life at the biochemical level contain massive amounts of digital information being used to produce and maintain an untold number of molecular machines and complex processes in every cell of your body every second. And the rest of my lecture is to show you a couple of examples of molecular machines. And we have videos for that. So it's worth staying because my time's up. For right now. Thank you. So I hope most of you can stay. I know if you have children it's probably hard, but I want to show you at least that last little piece, which we ought to talk about for the next week. But we'll get it in hopefully in about 10 minutes. Okay? Thank you for your good attention, class. I didn't see anybody sleeping. That's pretty good, because this is heavy stuff. So thank you for your attention. We'll come back to this after we have our closing. What do you think he's made of, class? Protein! He's a kinesin walker, and he walks on microtubules like this. He does this. And he carries stuff from one place to another. You think it's important in your body that something is transporting materials across the cell? It's got to happen. All kinds of things happening in there that need to be transported, maybe to another storage place or for other purposes. And kinesins are doing that all the time. So are dyneins. And there have to be microtubules, which, by the way, are made of? Yes, and have to be constructed. So they have to be built. So there's all kinds of stuff going on. And then dynein walkers are also very busy. So let's go on to the next one. This, ladies and gentlemen, is an example. We're going to look at a video of mitosis. You remember this from biology? Mitosis is cell division. And you probably are aware that cells are dividing all the time. I have some statistics here. Anybody want to take a guess at how many times in a day? You have about 30 trillion cells. You listening? You want to take a guess how many times a day one of those cells mitoses reproduces itself? Mitosis takes one cell and makes two, doesn't it? Exact copies. How many mitosis activities do you have in your body every day in your 30 trillion cells? You probably say a lot. The answer is, and I base this on a study in the magazine Humans on January 23, 2021 by Michael Steen, quoting biologists Ron Cinder and Ron Milo of Weizmann University of the Sciences in Israel. They used as a standard reference, a 20 to 30-year-old male, 154 pounds, 5'7". So you can figure out where you fit in that. In those conditions, that guy has 330 billion mitoses every day. That's 3.8 million per second. So what I'm about to show you here happens in your body 3.8 million times every second. You think that's important? If it weren't happening, you'd be dead. Now here are the percentages. 86% of those are blood cells. Most of them erythrocytes, that's red blood cells. Your body is making those in massive amounts every day. And if you didn't, you'd be dead. 12% are gastrointestinal epithelial cells. It's in your gut. 1.1% of them are skin cells. You do know, don't you, your skin is mostly dead skin? Your skin is made up of several layers. The new ones are formed at the base and they move their way up, and the further they move up, the deader they are. Sorry, girls. Your outer layers of skin are a bunch of dead stuff. I know you treat it all the way you can, and I love that, but that's what you got. How about 1.1% of 330 billion? That's 3.63 billion skin cells per day. So I tell the kids, take your bath. Did you hear back there, young man? You need to wash off those skin cells that are dead. That's 42,000 skin cells per second that are dying and mitosing. All right, I just thought that was interesting. So what I'm about to show you is very common in your body. Happens all the time. So let's go now. Hayden's going to be my guy. Here you're actually watching a picture of it. This is the nuclear material. This is the chromosomes. Stop right there just a second. See, what I haven't shown you, class, is the chromosomes. That bunch of chromosomes there have already copied themselves completely and made another copy up here. That whole process is extremely complicated. And we ought to show that to you. That's another whole video. But that's already happened. So you got the copy already made, and now it's going to start dividing. And let's watch it now. As the cellular material splits off, lots of stuff's happening in here that you can't see. And eventually there are poles formed, and they start separating. They're pulled apart, and then the walls divide, and you end up with two cells where you had one before. Isn't that amazing? Now we're going to start over. Here's the original process, but we're going to focus in now on one chromosome, this guy right here. This is a metaphase chromosome. Let's go up a little closer. Stop right there just a minute. Can you see there's 2 copies of this guy already? There's one, and there's one behind him. Everybody see that? So copies already been made. This is one chromosome, and we're going to watch it now as it splits. And what I want to show you is what are the stuff's happening down at the nano level that you don't see. So let's go. You see these? Those are microtubules. And this down here is the active site. It's called the kinetochore, the reddish area. It's not reddish, class. He does that for a reason. But it's the important part, where the activity. Stop right there just a second. It's a giant molecular machine that controls chromosome movement. You've got to move those chromosomes apart once they're copied to the poles. To make that happen, you need machines. And particularly cargo machines. Things has to happen. So that's what we're going to do. Focus down in here now. Let's go. Understand, I'm skipping a whole lot of things in here, not telling you everything. I'm telling you a few things. So we're going to focus in on the kinetochore. Let's go down in close now. First of all, can you see these microtubules are being built back here? What are they made of, class? Proteins that are being constructed. Stop right there just a second. Those microtubules run out here, way over here, and over here they're being torn down and recycled as fast as they were being made. Like any good government program, right? No, this is better than a government program because every piece of this is vitally important. Not only do you have to make it out of the proteins, and you have to make it properly or it won't work, but when you finish, you need to break it down so it can be used again properly. Our recycling programs are pitiful compared to what your body does. All right, let's keep going now. So the lots of, what do you think down in here, active and doing all kinds of work? Proteins. Am I telling you about all of those? No, I am not. But there are a bunch of them. In particular, there's some in here that are particularly important because they help time the process. It comes now to a certain time when it's time to split those things apart. Well, how do you time that? You use proteins. They know how to count, some of them. All right, so we're down close now, and watch, you're going to see it turn green. You see that? That means it's time to go, not because they really did turn green, but I want you to watch now. We're going to start seeing Dineen walkers. Here he goes. See him going? The Dineen walkers are heading out. And let's stop right there. Let's go one further. Watch this a little bit right there. Watch his feet. See those? So he's over here. Let me just illustrate here. He's doing this. Folks, I could tell you tonight, if you'd like, if you want to stay an extra 20 minutes, the exact chemistry that's happening that make this leg lift up and move forward, and then this leg moved up and move forward. It's just following chemical processes. But it's doing it on its own. And Dineen walkers are critically important to this process. They're taking stuff this direction. And if you watch closely, there's some other guys going the other direction. You see this little guy up here? That's a kinesin walker. He goes the other way. Dineens go this way, kinesins go that way. They both have vitally important things to do. Go. What do you think these are right here, class? They are proteins, stabilizing proteins that keep these microtubules in place. And then these guys are carrying globs of, what do you think? Proteins and other stuff as they are busily engaged in making things happen. Now we're going back up to the large size scale. All of that's taking place down here along these microtubules. And when the time is right, it turns green. Wrong. It starts separating. And what it does for one chromosome, it does for all of them. But there's a whole bunch of little machines doing all kinds of work in there in every one of those 330 billion cases. Okay? Isn't it interesting what came about by normal Darwinian processes, the blind forces of physics acted upon by natural selection? It's the most ridiculous fairy tale I've ever heard. All right, we're through with this one. Can you move it forward for me? There you go. I want now to take just the last few minutes and focus on the bacterial flagellum. It is the most amazing machine I know how to talk about. So I got to tell you a little bit about it. Bacteria, many of them have tails. They have little filaments that whip around like this, rotary tails. And we've learned how they work. And the reason we have is because one of the most important ones is E. coli. Tell me what you know about that. Yeah, if you get the wrong stuff, it'll kill you. E. coli. But there are strains of E. coli class that are required in your intestines or you're dead. You have millions of little E. coli swimming around in your intestines. Sorry, that's a little unpleasant. But the fact is you have an entire biome in your in your intestines. That's what you do for getting biblical, right? In your intestines that are not you, but they're living in there. And E. coli is one of them. And he's critically important to your existence. Does kind of all kinds of important things for you. And E. coli have flagella, filaments. And why have we studied them so much? Because E. coli is so critical, it can either kill you or keep you alive. It's very important to know what's going on. So we've studied that little guy for years. We know all about his flagellum and how it works. And I'm going to have a little movie here. Don't go anywhere yet. I want to read you a quote from a gentleman named Howard Berg, Harvard biophysicist who studies E. coli a lot and lots of other small things. Isn't that cute? That's a testimony to God, isn't it? Wow. Here's what Howard Berg says about E. coli. He says it's the most efficient machine in the universe. It uses almost no energy. Its motor rotates at 6,000 to 17,000 revolutions per minute, which is 283 revolutions per second. Watch. I can't even do 283 revolutions per second. Yeah, that's the truth. And the fastest motor vehicles we have produced don't get up to 17,000 RPM. The best racing machines, not anywhere close. But this little guy does it. And the filament that's attached to it can rotate 200 to 1,000 revolutions per minute. That's 17 times per second that filament goes around. And here's the remarkable thing, class. That filament, when it goes this way, drives the thing one direction. When it turns the other way, it drives it the other direction. And it can reverse directions instantaneously and be right up back to its speed again and use almost no energy. Man has never invented anything even close to this. Sorry, engineer. Not even close. But your body has millions of them swimming in you, doing it all the time. So let's take a look at him. Here we go. We're going to see some bacteria swimming. These are just regular type bacteria. I forget the names of those, but I'm not interested. So we're going to watch the next one come along here because he's a lot faster. See him go? Stop right there. These guys have at least 4 filaments. Some of them have up to 10. And what they do is they wrap around each other and then they flip. And so it makes you go fast. That's E. coli you're watching right there. And I'm sorry, but you're sitting there with millions of them in your right now. All right, so here we go. Keep going now. We're going to now make the video. It's a cartoon. You're coming up now with a cartoon. Here is the actual E. coli. Stop right there. And here are the 1, 2, 3, 4 filaments. You see them? And they come in here and they connect up through the cell wall. And this filament is what drives that little guy. It rotates. It's a rotating motor inside there that makes that happen. And what I want to do is show you what's happening there. So let's go. So here's where you're going to hook up. Here's the filament. And here's the hook. You see it? I'm going to stop there when it says the words. Hook, there's the filament. And there's the hook. Now stop it, okay? Now, can you see, class, that this hook is a different color? You know why he does that? Because it's a different protein. You see this connecting filament? It's a different color. That's a different protein. Here's another connecting that's a different color, and that's a different protein. And the filament is a different color. That's another protein. Right there, class, is 4 distinct proteins that each have their specific task, and they have to be right. And not only that, they have to be the right length and the right number of parts. This hook, class, I could read you exactly. I can't keep these numbers in my head. But there has to be an exact number of proteins in there that are exactly like 500 nanometers long, something like that. Don't hold me to that number. But it's got to be right. If it is not right, it doesn't work. And if you got a filament that doesn't work, guess what you got? A dead E. coli, which is probably good if it's a bad one. But there's lots of good ones. So let's keep going now. Now as you go down through the cell wall, you see all these rings here, and then down here's the basal body, and these things are called stators. Can you see how they're hooked to the middle wall here? They've got, and then in the middle is a rod. Stop right there just a second. So you've got rings, you've got the rod going up the middle, and you've got a layer that's inner, middle, and outer. And you've got rings. And this thing can turn up to 17,000 revolutions per minute. All right, let's go. Let's stop right there just a second. These things right here called the stators. Do you think if a motor is rotating at 17,000 revolutions per minute that it might shake a little bit? Folks, if you didn't stabilize it, it would rotate that cell to death in an instant. Every rotary motor that's ever been produced has stators that stabilize it. That's what these do. Only this is just there automatically. Somebody did that, class. Let's keep going. There's the stator that I just talked about. It goes all the way around. You know, this is cut across. It goes all the way around. There's the rotor. Now, stop right there. This is my last point about the flagellar motor. When that little bacteria divides, where one cell becomes 2, it does not reproduce the flagellum in the division process. So it has to make them again. You listening to me? We know because we've watched it. It divides and makes 2, right? You got 2 bacteria now, but they don't have any flagellum. So from the inside out, they build the flagellum, each one of them. And they build them in the precise order as if you were building a tower on the campus of a university. Where do you start? You dig a hole in the ground and make a foundation, right? And you make it solid and everything has to go in there and everything has to be right before you ever start anything else, isn't that right? Then you have to bring in the right stuff to do the next piece, and then the next piece, and right on up, and then you add the electric and you add whatever, plumbing and all the other stuff that goes with it, and then you go up to the top. But everything has to be done in place, in order. An engineer's job is to orchestrate that so it does exactly what it should in the order that it should, right? There are no engineers in this thing. But what this Japanese company did is they examined every piece of that process, and we're going to show you how it works. And I'm not going to give you anywhere near all the detail. But let's go. Here's the rebuilding of flagellum. It takes about 20 minutes in nature for E. coli to build its flagellum back. The first thing that happens is the fly-F protein. It has to be constructed. It's called the MS ring. And there's an exact number of those. And then the fly G and the fly M come up next. No, the fly G is next. And then the fly M and fly N. Those in order. Stop right there just a second. Those are different colors. Why do you think, class? Say it louder. Different what? Yeah, different proteins. and there's a precise number of each one of those every time. And it's built in that order. Not different. Keep going. Now you've got a rod that comes right up through the middle. And that thing is absolutely incredible. You notice it's a different color. That's a different what? Protein. And then the top of it is called a cap. That's a different color. It's another protein. And part of the purpose, stop right there just a moment. It's also building the stator in the process. We're not even talking about that. That's another bunch of proteins. But this thing right here has got to drill holes through that wall, class. You think it's healthy for an E. coli to drill holes through his wall? No, that'll kill him. So what happens in the process? As that drill goes up through here, you watch. They're going to try to illustrate what's happened. Go. I don't know what's going on there, but it sounds like a drill, doesn't it? Okay, see the hole forming? Do you notice there's a ring forming right instantaneous? Stop. Those two rings, that's the L ring and the P ring, formed at exactly the same instant. So the hole is stopped up and the thing is held. It just does it. And by the way, class, most of those proteins come up through the rod and are shoved up. Not these guys. They come in from the outside. These two proteins have to be brought up by what are called chaperone proteins. Exactly. Which are not up here. Chaperones. In other words, this guy would lose his way if he didn't have a chaperone. Bring him and place him in place exactly where he needs to be at the time he needs to be there. I do this. You tell me, brilliant scientist, that happened by Darwinian evolution, the blind forces of physics, over millions of years, in some small steps. It's a fairy tale. Keep going. There's the L and the P ring I mentioned. Now look, after that thing's done, you've got to make the hook cap. It has to be exactly right. It's 5 pieces, and it builds that hook. Stop right there, yeah. That hook has to be exactly precise. And when it's done, that hook says, I'm done with my job. It's released into the cytoplasm and recycled for use again. And then, let's watch. As the hook is, you get that intersection, that junction protein and the other junction protein, and then you got another cap. Stop right there. Can you see there's 1, 2, 3, 4, 5 legs to this thing? This one is so important and has been studied so much, it's given a name. She is called Twinkle Toes. I don't have 5 legs, but watch. The 5 legs are dealing with 11 holes. So you can step around from one to another like this. And as that thing lifts its leg and moves around, then the filament, can you see those? Well, you can't see it yet. Let's go a little bit further. See the protein's coming up through the bottom here? And it comes out. Now, this leg is in the right position in that one that can go out this way. You see that? Go ahead. The next one comes out this way and then that way. Can you see it? Those legs are stopped. Those legs are forcing those filament proteins to go out in the right order and form this amazingly beautiful thing that interlocks with itself with a hole in the middle. It's important to have that hole in the middle because it needs to be flexible, but it also needs to be strong. And all of that's happening at the same time. And so little Twinkle Toes is orchestrating that like this. Let's keep going. And you watch her a little bit. Of course, that's going way too slow. But he'll speed it up in a minute. See her Twinkle Toes? Except it goes way faster than that. So let's give them some idea. Much better. Isn't that glorious? And the average filament like that has filament units up to 32,000 units to make the filament. I told you I want you stupefied. when you leave. That class is in one little creature. If you turn your thumb sideways and look at your thumbnail sideways, you could stack 1,000 of those guys across your thumbnail. That's how tiny they are. You listening? And they're swimming around inside your intestines all the time to keep you alive with filaments like this that are glorious. It's the most wonderful machine in the universe, put in a little one-celled organism by the God of heaven, not by evolution. So I close by saying those are multiple protein processes. Each protein is complex, suited for its function. Each machine is highly sophisticated, intricate, dynamic, and elegant, and these machines are spontaneously self-assembled in the cell class. Not only does that little one-celled organism have that amazing machine, it built it like the Ford factory with no engineers. That was designed class. And there are thousands of them in every living cell, thousands of them. We haven't touched the hem of the garment. I'm closing by introducing you to Dr. James Tour, Professor of Chemistry, Materials Science, and Nanoengineering at Rice University. He's one of the world-renowned nanoengineers. He's dealing at the nano level a lot. Marilyn and I were privileged to meet him. I did a meeting in Houston, and he lives at Rice. We got to meet him. He was quite a guy, wasn't he? And one of the things he did when we got through was he asked if he could lead us in a prayer. And it was a beautiful prayer. He's a Messianic Jew. He's a Jew, but he believes Jesus is the Son of God. And if you go to his website, he will do his best to convince you, if you want to become a Christian, you have to believe in the resurrection of Jesus, and here's why you should. Reminds you of Paul on Mars Hill. Anyway, I love this man because he is a brilliant scientist and a strong believer in God. And he talks about how such foolishness when people talk about how this happened by natural causes. He does all kinds of work with nano things like graphene nano ribbons. That's carbon, one carbon layer thick. How do you get that? That's hard. We've learned how to produce it, and a lot of things use it for. One of the things he did with the Congress was to tell them you can use this stuff to help produce better concrete. Put carbon graphene in it, and you can produce better concrete. And you know, one of the places you can get carbon graphene from